Hi. Now we will discuss about, in terms of engineering aspects, how to manufacture monoclonal antibodies and differences between monoclonal and polyclonal antibodies. So how to manufacture monoclonal antibody for clinical use. So we can use antibody for clinical therapy. So for that, we want to first understand antibodies are produced by the B cell or B lymphocytes inside our body. So for example, we can collect antibody from patients. So patients who have high titers of or high concentration of antibody specific for a particular antigen, let's say such as there's a very special infectious disease and we want to collect the serum of the patients and to find out that antibody as a treatment. So for example, if there's a hepatitis B as an antigen, then antibody against hepatitis B surface antigen, writing down HBsAg, and we can collect the serum of these patients and isolate those antibodies from this pooled plasma. And one of the risks is not only during this isolation, that carries the risk of other kinds of infectious disease can transmit. For example, like HIV, which will be a big problem. So this is one way to collect and gather antibodies. So another way is we can use animal. This is more typical. So such as mice or rabbit or goat. When you are purchasing antibody for experiment, you will find out these species. So how do we manufacture antibody? You know, antibodies are pretty expensive, and the reason is here because when we generate, we are intentionally exposing the animal to a specific antigen of interest or immunizing the animal, such as, let's say, hepatitis B surface antigen, we expose this one to those animals. And then we wait some time and we collect blood serum from that animals. And think about it, what kind of antibodies against these antigen will be collected? Then we have to consider about the shape of this antigen. Let's say these are the hepatitis B virus. Then this virus will have a different kind of a antigen or epitopes, different kind of epitopes. So these are as a foreign getting into the animal's immune system will develop antibody against these antigen, especially you can recognize there are multiple sites of epitope. So the blood serum from animal will contain not only single antibody against, but multiple. So that we call a polyclonal antibody. Because why do we say clonal is this antibody will be produced by many different B cell clones. Clone means let's say there is a very specific antigen against the antibody. The B cell will create clones of these the same antibodies, while another B cell will have another epitope will generate another clones of antibody. So this will cause differences in this polyclonal antibody will have differences in epitope, or because its epitopes are different, so each of these will have different affinity against the administered antigen. So what will be an issue from here compared to from patient? From patients, the antibodies are human antibody, while from animals, we can see the antibody, heavy and light chain, constant region will be from animal. and that it's in fact foreign to the patient for clinical use. So the caveat here is foreign antibodies can cause immune reactions. So it may be limiting treatment to a single exposure, because one single exposure will cause the human body create lots of antibody against this animal antibody, so that it may cause a immune reaction next time. So this is a diagram showing manufacturing antibodies, the effect. So x-axis is time as a week, y-axis is a logarithmic scale of antibody concentration in serum. So initial exposure will cause the B cell, specific B cell to produce antibodies. So initial antibody on the surface of the B cell will create IgM. And then over time, IgG, the amount will be exponentially increased. And you can see that after injecting antigen A. And 6 weeks after, let's assume that we inject antigen A again. So what happens for this antibody against response to A is IgG is naturally decreased, and then this second exposure, it will boost up again. But you look at this slope is different. That is, the second time is much faster, because our body immune system will recognize this antigen A and be ready. The next exposure, it will be much faster to respond. It's because it's called the memory effect, because we remember what happened before. While if we happen to inject antigen A and B together at week of 6, the IgG or immunoglobulin G antibody against the B, the response will be not as stiff as this is the first exposure against antigen B. So the memory effect can be explained as specific antibodies are produced after exposure to antigens. So now the previous ones are polyclonal antibody, but in many cases, monoclonal antibody will be more useful. So for example, to detect or purify a specific substance, we better to choose a monoclonal antibody. And this become a very important tool in biochemistry, molecular biology, and medicine to prevent, diagnose, and treating of disease. So let me show you the scheme how to create monoclonal antibody. This is so clever and important that it leads to Nobel Prize in 1984. So let's take a look. So the mouse will be challenged with an antigen. It can be a pathogen, and then the spleen contains a lot of B cells, but the B cells is hard to culture for a long time. So his idea is, hey, how about making a B cell cancer called the myeloma cells? The cancer cells will proliferate indefinitely. So how about fuse these two cells together, and these fusion ones, you isolate a single cell, which is successfully fused one, then we culture in a medium selective for this positive cell. Then it's a kind of cancer cell, so it keeps proliferating. So you can have a lot of the same cell clones, and this will produce exactly the same antibody or monoclonal antibody. And that monoclonal antibody, we write it as mAbs, and this is a monospecific antibodies produced by identical cells, because these cells are now clones from the cancer characteristic. So that monoclonal antibody will have exactly the same chemical structure, and antigen specificity is exactly the same because the same epitope. And of course, then the antigen binding affinity will be also the same. So this kind of fusion of a specific antibody producing B cell with a myeloma cell or B cell cancer, and that idea, those hybrid cells we call as a hybrid, and so oma means cancer, so it's called hybridoma cells. And like drug names, you can actually recognize which one's a monoclonal antibody. Because for example, as I told you multiple times, Herceptin, which is anti-HER2 antibody drug, its name is trastuzumab. And you look at this little final map, it means it's monoclonal antibody treatment drug. Okay, you can watch this YouTube for explaining monoclonal antibody. And now I want to discuss about humanized antibody. Because once we create this monoclonal antibody, but this monoclonal antibody, the Y shape will be produced in the animal. So it's an animal antibody, which means it has a variable region and constant region, and that itself is not present in our human. So once you put it inside the human, we also can develop antibody against this foreign antibody. So it may be not easy to use as a drug because it's once inside our circulation, we may attack this antibody cell. So the idea is, can we make, except the variable antigen binding sites, the rest of them, can we make it as a human-like or humanized so that it doesn't cause immune reaction from our body? So what is humanized antibody? Monoclonal antibody. That's antibody from non-human species, which is potentially immunogenic to human, and whose protein sequences have been modified to increase their similarity to antibody variants produced naturally in humans. So this is a kind of cartoon showing that. So murine, the yellow means murine. So if this one is inside the human body for clinical use, that can cause a problem. So we create humanized or chimeric antibody. So with monoclonal antibody to administer to human for clinical use, and that we call as a Zumab. So remember, I just discussed trastuzumab as a Herceptin or anti-HER2 humanized antibody, because there's now you see Zumab. That Zu means humanized. And how do we produce this? We better to see a little more detail using recombinant DNA technology to create constructs capable of expression in mammalian cell culture. So recombinant DNA technology is very, very important. So let's take a look further. So this blue means a mouse, and red, this brown means human. So the top row shows some pure mouse, and this is chimeric because that FC region is now replaced into human ones. So that's chimeric. And even these antigen binding sites, some part can be replaced as humanized. So this is humanized. And even chimeric humanized is existing, that's human. So what are the advantages of this chimeric and humanized antibody? Because now we have this one, it will have a good biological activity of the binding. And because this body is humanized, it will reduce immunogenicity. And because of that, inside circulation, it will last longer in the circulation. So that means it improves pharmacokinetics because of the prolonged plasma half-life. And these chimeric antibody now as a name, it's put C. So CMAP means it's a chimeric antibody. So one example is called Erbitux or anti-EGFR chimeric antibody. EGFR stands for epidermal growth factor receptor. And you see that drug name is cetuximab. Now you read this, or mAb is monoclonal antibody, cMAb, the C means chimeric antibody. Okay, so next time we will discuss about vaccines and viral infection and the requirement for effective vaccine development. Thank you for your attention.